Lesson 3J, Factors Affecting Enzyme Function, Rate of Reaction. There are many factors that influence enzyme function. The first is substrate concentration. If you have a set amount of enzymes, then the reaction can proceed quicker if more substrate is provided. With low substrate concentration, the likelihood of an enzyme binding with the substrate to form the necessary enzyme-substrate complex is low. It's simply more difficult for the enzyme to find the substrates when there's less of them. In this picture, the green enzymes must bind the yellow substrate. As you can see, if we add more substrate, it means there's a better chance that the enzyme will bind the free-floating substrates. It's even more apparent with more yellow substrates. This graph shows the effect of increasing substrate concentration on the rate at which the products are formed by the enzymes. This is also known as rate of reaction, ROR. We can see that the ROR increases as the substrate concentration increases, allowing for the enzymes to work faster and faster. However, there is an upper limit to this rate, as the enzymes will eventually reach their top speed and not be able to proceed any faster, even with unlimited substrate. This is the plateauing of the graph. The second effect on rate of reaction is enzyme concentration. If we have unlimited substrate, then the enzymes can only work as fast as one can bind the substrate, complete the chemical reaction, and then release the products. This means that each enzyme has a maximum speed for catalyzing a reaction. In this picture, 2 enzymes working at max speed cannot match 6 enzymes. And 12 enzymes would obviously have a greater rate of reaction than both earlier scenarios. The graph for this would look similar to increasing substrate concentration. The ROR will increase as more enzyme is added. This will increase indefinitely if there is unlimited substrate, but if there is limited substrate, then the ROR will come to a complete stop once the substrate is completely used up. The third factor is temperature. Most human enzymes function best at 37 degrees Celsius or so. Too warm and protein physically changes, and we know that protein shape is very important to its proper functioning. This is called denaturing, and this process can easily be seen when a chicken egg is cooked. The protein of the egg white becomes denatured. If a protein is denatured due to high heat, the process to renature it is almost impossible. Slight temperature changes may have no lasting effect though. When a protein becomes too cold, it loses flexibility, which restricts its ability to conform to the shape of the substrate, as we saw in the induced fit model, and it will become deactivated. Unlike denaturing, deactivating is reversible in most cases, and simply warming up the protein can reactivate it to full function. This graph shows the optimal temperature for human enzyme function. Anywhere around 37 degrees Celsius is fine, but too cold and the rate of reaction falls off, as the protein enzyme is deactivated. High temperature to the right also has the same effect on rate of reaction, but high temperature will denature a protein, which is irreversible. pH is also concerned for protein function and rate of reaction. Acids and bases both have a denaturing effect on enzymes if the pH strays too far from the optimal pH. In fact, this egg has been cooked using strong acids. Unlike temperature though, pH in the human body varies from organ to organ. pH of the stomach is pH 1, pH of the blood is 7.4, and optimal pH of the small intestine is roughly 8. This graph shows 2 human enzymes, pepsin, which helps with the chemical digestion of proteins in the stomach, and trypsin, another enzyme for digestion of proteins but located in the small intestine. Each enzyme has an optimal pH, and if you stray from it either too basic or too acidic, the enzyme will become irreversibly denatured and will decrease the rate of reaction. The fifth factor is enzyme inhibitors. An enzyme inhibitor is a molecule that prevents an enzyme from functioning at its optimal. There are 2 types of inhibition. One is competitive inhibition, where the inhibitor molecule binds to the active site, preventing the proper substrate from binding. In this example, you can see the green inhibitor has bound to the active site of the gray enzyme. This prevents the yellow substrate from binding to the enzyme. Therefore, the chemical reaction is halted. An example of this is the poison cyanide, which inhibits the synthesis of ATP for energy production. And the second is non-competitive inhibitors, where a molecule binds to some other area of the enzyme other than the active site, causing a physical change to the enzyme. We call this a conformational change. We know that the final folded shape of a protein is important for its function, so a conformational change caused by an inhibitor can greatly affect the enzyme's function. In this example, the pink inhibitor has bound to a different site on the gray enzyme, which has resulted in a conformational change of the active site. This prevents the yellow substrate from binding, or it may prevent the enzyme from completing its chemical reaction. An example of this type of inhibition is penicillin, which inhibits bacterial wall formation. These types of inhibition can be further divided into reversible and irreversible. Reversible inhibitors will bind to an enzyme and then release them, allowing for the proper substrate to bind on occasion, which is similar to deactivating the enzyme, which can be reactivated later. On the other hand, once irreversible inhibitors have bound to the enzyme, they will not release, which essentially deactivates the enzyme permanently. On this graph, the green line shows the rate of reaction of an uninhibited chemical reaction. The blue line shows the effect of a reversible competitive inhibitor on ROR. You can see the rate of reaction is slowed until an ideal amount of substrate is available to outcompete the inhibitor for the active site. The red line shows the effect that an irreversible non-competitive inhibitor has on ROR. As you can see, it will plateau as it reaches a max rate of reaction, well below the non-inhibited reaction. This is due to the number of enzymes that have been permanently deactivated by the available inhibitors. The last factor is the absence of cofactors or coenzymes. As you'll recall from the last lesson, these are molecules that are necessary to complete the enzyme substrate complex so that the catalyzed chemical reaction can occur.